DNA can be represented by two anti-parallel strands, these two here. When DNA replication begins, a replication bubble forms in the heart of the nucleus. At specific sites called origins of replication, the double-stranded DNA begins to unwind, like over here. A helicase here, it glides along the strands and breaks the hydrogen. And breaks the hydrogen bonds between base pairs, AT and GC. Unzipping the DNA double helix here. So the strands open, revealing their nucleotides. Template strands are revealed and waited to be copied. Primase lays down RNA primer. This primer marks the starting point for DNA synthesis. A leading strand glides smoothly along the template with the DNA polymerase partner adding nucleotides in a continuous stream, like down this way. The lagging strand, which is down here, the lag strand, follows and creates Okazaki fragments, like, over here is one, Okazaki, and up here, well, they're both close to each other, these two right here. Its DNA polymerase stitches these fragments together. It then stitches these areas together here. And ligase weaves phosphodiester bonds, sealing the gaps. Here. The strands move in opposite directions. The directionality of replication occurs as the leading strand glides forward. This is the leading strand up here. Leading strand. And the lagging strand moves back and the replication fork expands. Here's the replication fork. The DNA strands complete their replication and create replication form, two identical DNA molecules. This is the process of transcription. So the circle in the back is RNA polymerase, and all RNA polymerase reads one side of DNA to make complementary mRNA strands. So that's this one, and okay. This strand down here is called the antisense strand, and this strand is called the sense strand. In addition, also DNA has deoxyribose while RNA has ribose. Introns are non-coding sections of an RNA transcript of the DNA encoding it that are spliced out before the RNA molecule is translated into a protein. The sections of DNA or RNA that code for proteins are called exons. The direction of transcription is to the right, the 5' to the 3. And then this is translation, and in the back is the ribosome, and it attaches to the mRNA as well as the tRNA with the corresponding amino acid and the codon codes for it and brings anticodons. So anticodons are the ones down here, and these chains are called the tRNA. And these ones, the CC, are called mRNA. Oh, yeah, right there. And then mRNA, the bases are grouped into threes and they create codons, which each are 4 amino acids, or each 3 codes for an amino acid. And the amino acids are connected by peptide bond and stops until it reaches the final codon, which is this one. And this is called a growing peptide chain because these are like peptide bonds, and they they're connected to the mRNA, and yeah.
The DNA, we're zooming in. The DNA opens back up.
Oh, I think she's talking to Kai.
Oh, stop talk.
Here comes the helicase. The helicase jumps onto the 5 end and opens it up. That's the replication fork. Here's a primase. Primase brings in a primer. And the DNA polymerase brings in a DNA strand, and that would be called the leading strand. The primase in the bottom strand, lagging strand, brings in chunks of primer,